In mammalian cells, the endoplasmic reticulum, or ER, plays an important role in protein biogenesis and calcium signaling. The heterotrimeric Sec61 complex in the ER membrane provides an aqueous path for newly synthesized polypeptides into the lumen of the ER. Recent data suggest that Sec61 may also form transient calcium leak channels. To directly address the role of the Sec61 complex as a potential calcium leak channel and to characterize its putative regulatory mechanisms, cells are first treated with siRNAs directed against the Sec61A1 gene. In complementation analysis, the cells are co-transfected with an iris GFP vector that allows for the siRNA-resistant expression of the wild-type Sec61A1 gene. Then the cells are loaded with the ratiometric calcium indicator, FURO2, and changes in the cytosolic calcium concentration are monitored via fluorescence microscopy. As the calcium levels are measured, solution changes are performed to assess the impact of various agents, such as calmodulin antagonists and thapsigargan, on calcium leakage. Quantitative analysis of the resulting images reveals changes in calcium efflux induced by the silencing of Sec61A1. In this video, a model for the role of Sec61A1 in calcium efflux will be presented following demonstration of the procedure and presentation of the results.
The main advantage of this technique over existing methods like planar lipid bilayer experiments or single-channel measurements is that candidate membrane proteins can be analyzed in their native environment and without tedious purification and typically inefficient functional reconstitution.
This method can help to answer key questions in the calcium signaling field, such as, for instance, what is the contribution of a specific